Today we start with linkage, and we will go back to this idea that we can predict a 9331 phenotypic distribution when we're dealing with two loci that are dope, fully dominant, penetrant, and expressive. And there were two other conditions. One was that the characters do not interfere with one another. That's what we discussed last week. When there is genetic interactions or epistasis, the genotype of one locus can affect the phenotypic manifestation of another one. You can see or not what's going on with a second locus if there is epistasis. And what we'll discuss today is what happens when the characters in question, let's say these two genes that we're working with, do not segregate their alleles independently from one another into gametes when making gametes for reproduction. So if I can take you back to the textbook example of a dihybrid cross that will give rise to a 9331. We mentioned it in class, the Mendel's P's, and how if you're looking at, say, P color and P skin, two characters that segregate their alleles independently from one another, that show full dominance, and that do not interact or interfere with one another, Mendel established that alleles for different characters could be inherited independently. And this is a consequence of these alleles being sorted into gametes independently from one another by contrasting observations to predictions. And what we mean by that is he knew that the phenotypic distribution for one character, let's say P color, was 3-to-1. 3 dominants for every 1 recessive, in this case 3 yellows to 1 green, right? And the phenotypic distribution for the other character was also 3-to-1. In this case, 3 round or smooth for every 1 wrinkle. And his reasoning was, if these are the outcomes with those distributions that I can get from these two independent events, if I were to calculate or predict the distribution of outcomes from both events combined, I can expand the binomial, and I expect to see 9 times both dominant traits, 3 times one recessive trait and the other one dominant, 3 other times the opposite, and only 1 time he expected to see both recessives. And that came from applying FOIL to 3 plus 1 times 3 plus 1, so you have 3 times 3, 9, 3 times 1, 3, 1 times 3, 3, and 1 times 1, 1, right? So you get the 9 to 3 to 3 to 1. So Mendel treated the phenotypic distributions of each of these characters as being inherited independently from one another. And it worked. The experimental observations matched the predictions. We also, by the way, this we discussed is due to the fact that the dihybrid individual is making gametes and sorting alleles for those characters into them independently from each other, which gives rise to all possible allelic combinations with equal probability. And because these individuals are making all kinds of gametes, all possible gametes with equal probability, you end up with a Punnett square that has 4 columns, 4 rows. And if you solve that Punnett square, you basically derive the same thing that Mendel was deriving by expanding the binomial. You can solve it by Punnett square as well. The expansion of the binomial is what we normally refer to as the probabilities method, whereas this is the Punnett square approach. There was another way of checking that the alleles for different characters were going into gametes independently, or in other words, without any regard for what other alleles from other characters were doing. And that was through, if you guys remember, through a test cross, right? So if we took that dihybrid individual from the F1 that was heterozygote for both loci, and we crossed it to a very special individual that is homozygote recessive for both characters, in this case it would be plants that produce green wrinkled peas, then what we would be doing is we would let the double homozygote recessive produce only one kind of gamete that has nothing but recessive alleles in them, allowing us to reveal what is going on with the other gametes produced by the dihybrid. And what that two loci test cross would reveal is that now there is a 1 to 1 to 1 to 1 phenotypic distribution, right? You don't have a 9 to 3 to 3 to 1 anymore because you're not crossing two dihybrids to one another. You're crossing one dihybrid to a very special individual that is homozygous recessive for both. And what this two loci test cross is revealing is, once again, the idea that Mendel deduced from his observations, and that is that characters will segregate their alleles into gametes independently from what other characters do. Right? I want to introduce here a new concept that is important for what we'll discuss later today, and perhaps even more so on Thursday, and that is the concept of parental versus recombinant gametes. This is a concept that made a lot of sense historically when people were doing a lot of these experiments, crossing parental pure breeding lines to generate some type of hybrid individual for 1, 2, or more loci, right? And then testing those hybrid generations against pure homozygous recessives or against each other in dihybrid, trihybrid crosses, right? And that was the concept of parental versus recombinant gametes. And the idea is that the gametes that the parental individuals produce, if they're pure breeding, they're going to be of one kind only, right? What defines a pure breeding organism is that they will always produce gametes that are the same. And that's why all of their progeny is always the same. Right? That's what defines a pure breeding line. But these hybrid individuals were producing all kinds of gametes. So the concept of parental and recombinant gametes relates to the idea that some of the gametes that these hybrid individuals produce are going to be like the ones produced by the parents. And some of them are going to be new, different. We call them recombinant because they tend to recombine, reshuffle, combine in different ways the traits that came from the parents. So let me show you. When we did the hybrid test cross, the two loci test cross with the hybrid, we got all kinds of recombinant phenotypes in a 1-1-1 ratio, 1 to 1 to 1 to 1 to 1 ratio, right? If we did the same with if we test cross the parents to the double homozygous recessives, what we would see is that in this case, the first pure breeding parent that happened to be a double recessive with green peas that are wrinkled, we cross it to the double recessive with green and wrinkled peas. Now, everything we get is wrinkled pea plants, right? Green and wrinkled pea plants, right? Whereas the other parental line that was pure breeding with double dominant would produce nothing but the double dominant, yellow and smooth. This is what basically showing graphically what I was saying before. Parental lines produce parental gametes of one kind. And we can reveal that through a test cross. There's going to be only one phenotypic class, and all of the progeny are going to look the same. This defines the types of gametes that the parents produce, and we call those parental gametes. In everyday parlor, it's almost like saying the genes from your mom, the genes from your dad. You know when people talk like that and they say, oh, those genes are from your mom, those genes are from your dad? Same idea here, that any hybrid individual will have inherited genetic information that came from one parent and some genetic information, half genetic information that came from one parent, half that came from the other, right? And those would have been carried by what we call the parental gametes with parental allelic combinations. But then these hybrid individuals can, at times, recombine, reshuffle these alleles that they inherited from each of the parents into new allelic combinations that we call recombinant gametes, okay? So once again, if we test cross the hybrid F1 from this particular cross to a double homozygous recessive, we are going to get all kinds of phenotypes in the F2, and those are due to different allelic combinations. Two of them were present in the parental generation, right? The double dominant and the double recessive, and we call those the parental gametes. But then these two other kinds of gametes are new. The parents, neither of the parents could make gametes like this. They just couldn't. So these are new, these are recombinant gametes. So far, so good? Yeah? The reason I need to introduce parental and recombinant is because now, from here on out, we will refer to them a lot. And essentially, it's always the same idea. What ability has an organism to create new allelic combinations when it's making its own gametes? Because that's going to give us a sense of how close together two loci are in, now we know, DNA, or how far apart they are in DNA. Because the farther apart they are, the more independently they're going to behave from one another, whereas the closer they are, the more they're going to go together everywhere they go. Did that make sense? So, if we reformulate Mendel's second law, but using this new terminology, what we can say is that when loci follow Mendel's second law, then these dihybrids will produce parental and recombinant gametes in a 50-50 ratio. Right? Half of the gametes that are dihybrid for characters that follow Mendel's second law will produce half of the gametes are going to be like the parental, half of them are going to be new. They're going to be recombinant. So we're basically rephrasing things that we said before, that when loci follow Mendel's second law, they're going to be segregating their alleles independently from what others do into gametes. From a parental versus recombinant gametes perspective, what that means is that they're going to be producing parental gametes or new combinations in recombinant gametes with a 50-50 ratio. Half of them are going to be like the parental gametes that gave rise to them, and half of them are going to be completely new allelic combinations in gametes that neither of the parents could produce. Okay? Cool. That isn't always the case, right? So for instance, there could be an example. We're going to go through this example here. It's based, I don't know the details, but it's based on historical findings, because after Mendel, when Mendel's work was rediscovered, everybody jumped into it and everybody wanted to try with their plants, their animals, right? So people started doing experiments like Mendel's all over the place, and they started bumping into exceptions to some of the observations that Mendel had made. For instance, someone was crossing these two types of wheat, I think it was, or rice, I can't remember. They had, once again, generated two pure breeding lines. One was double recessive with long grains and yellowish, and the other one was double dominant with thicker, I guess, more oblong grains and more rounder grains and brown. And knowing that the double recessive was the long and yellow, they test cross the dihybrids, expecting to see what you would see if these two characters followed Mendel's second law. That is, that there would be half of the progeny reproducing the parental phenotypes, the double recessive and the double dominant, and that the other half would be some recombination of this. For instance, long and brown, and round and yellow. But those two were actually missing. The recombinant phenotypes were missing. Okay? So what these observations reveal, and this started happening more and more, that people started noticing, is that sometimes loci don't have the freedom to assort alleles independently from each other into the gametes. When that happens, we say that those loci are linked. And intuitively, what this means is that if in a parental line you had a recessive for one and a recessive for the other, then from that point on, those two recessives will go everywhere together. And if in the other parent you had a dominant for one and a dominant for the other, same thing. Those two dominants would travel everywhere together. They're linked. So when it came the time for these dihybrid individual to produce gametes, it did not have the freedom to assort alleles for each of these two loci independently into the gametes. The dominants always went together, into half of the gametes, and the recessives always went together into the other half of the gametes. In other words, when you have linked loci, hybrid individuals, dihybrid, trihybrid, whatever, whenever the loci are linked, whenever they're fully linked, there's going to be 0% of recombinant gametes, and all of the gametes are going to be like the parental. This dihybrid here is producing half of the gametes like one of the parents produced, and the other half of the gametes like the other parent produced. Notice they're not all of the gametes are the same. These hybrids are not pure breeding lines, okay? So it's not that they're producing all identical gametes. What I'm saying is that 100% of those gametes are going to be like the parentals. Half of them are going to be like the gametes that mom produced, mama plant, I guess. And then the other half are going to be like gametes that daddy plant produced. You're laughing? I'm going to be looking at you. So full linkage means that the alleles for a given locus are always traveling together with the alleles of a fully linked locus. And they're going into gametes and to the next generation together. So here's just some text repetition of what I just said. So when there is no linkage, meaning when we have full Mendelian inheritance, half of the gametes produced by a dihybrid, or in general a hybrid individual, are like the parental, and half are recombinant. When there is full linkage, the other extreme, all of the gametes are like parental, no recombinant gametes whatsoever. Notice that what I just said relates to a specific pair of loci, right? You can have two loci that are fully linked, and either of them completely not linked to a third one, right? So the SB loci that we were discussing in the previous slide don't have to be linked. They're linked in between them, but they don't have to be linked to a third locus, let's say locus A. Right? So all of this business about being fully linked or not, or being completely unlinked, and producing parental or recombinant gametes always refers to a specific pair of loci, or 3 loci, or 4 loci, however many loci you're dealing with, okay? It does not really refer to all of the gametes produced by the individual. Because, once again, like we said several times by now, the gametes, there's no 2 gametes that are the same produced by an organism, because it's all kinds of reshuffling of genetic information that happens due to random sorting of the homologous pairs of chromosomes on both sides of the equatorial plate, crossing over, all of those sources of variability that will introduce a lot of diversity across all gametes. But when we're talking about 2 particular genes or loci that are fully linked to one another, then the alleles for those are going to travel together no matter what. I know some of these may look like Mendel's plants. They were not. It's a different species. These experiments were done early 1900s. And they show something peculiar. So what the scientists behind this experiment did was cross, as usual, 2 pure breeding lines. And these were lines that were pure breeding for 2 characters, flower color and pollen shape, right? So I'm going to zoom in a little. The first pure breeding line produced purple flowers and long pollen grains. The second pure breeding line produced red flowers and round pollen grains, right? These were pure breeding. So anytime that you cross these plants to themselves, you get nothing but purple flowers and long pollen, or red flowers and round pollen. So far, so good? We cross them to one another, and we get an F1 that is 100% purple flowers and long pollen grains. So what were the dominant traits for these 2 characters? Purple flower and long pollen grain, right? So purple flower fully dominating over red, long grain fully dominating over round pollen. Okay? Then we do the usual. We do a self-fertilization. And let's say that through a lot of these self-fertilizations with the that trick of taking pollen grains with a brush and sticking them in the ovary of the flower, and you have a large garden and you do it several times and you get a progeny, very large progeny of 380. My question to you, and I'm going to give you a couple minutes to think about this, what would be the phenotypic distributions, what would be the phenotypes and in what ratios, relative ratios, if these two characters were not linked at all or fully linked? So what are we thinking? Let's start with the, go ahead.
So if it's not linked, I think it would be the 9-3-3.
9-3-3-1,
It's not directly from.
Okay, so let me unpack that. So the argument here is if they're not linked at all, you expect a 9-3 to 3-1 ratio. That's what you would expect by classic Mendelian genetics, right? Classic distribution of an F2 from a dihybrid cross following Mendel's second law. If they're fully linked, the argument here is it would be a 3-1 because the dominant, what did you say about the purple flowers and the red flowers?
So the dominant would be purple flowering long pollen, and it would just stick.
Oh, stick together. So the argument here is, remember how Mendel said, if these characters are behaving independently, then I have a 3-1 for one and a 3-1 for the other. I treat them as independent events. And if I want to calculate the combined probability of them happening together, I apply FOIL and I get 9-3-3-1. But what you are saying is that no, these dominants are going together, so I will combine these two threes into one. And then I will combine these two ones into another one. Did I follow your argument more or less correctly? Yeah? Any other ideas? I overheard some 1 to 2s to 1s. There is one way in which the 1 to 2 to 1 can happen, and that's where I was saying, I hope I remember to say before. So if instead of having the two dominants in one parental plant and the two recessives in the other, we would have had one of each, the phenotypic distribution in the F2 would not be a 3-1, it would be a 1 to 2-1. I invite you guys to try that at home after we go through this example here. And you apply it to when you have one dominant and one recessive, and one recessive, one dominant in the other plant, okay? All right, so there's two ways to approach this. You applied the probabilistic approach. You said, you reasoned like Mendel in a way. You said, I have a 3-1 for one and 3-1 for the other, but now they're together. So when I say 3-1, I don't talk about 3-1 of one dominant over the recessive. I'm talking about a 3-1 of both dominants to a 1 of both recessives, right? That sounds reasonable. The other approach was to do Punnett squares, right? So if we do Punnett squares, that's solved here. If the loci are not linked at all, the dihybrid plant will be putting alleles into gametes independently from what happens in other loci, so it could come up with all kinds of allelic combinations. It would produce the two parental gametes, but it would also produce the two recombinant gametes. And because each of these gametes has equal probability, you end up with an even Punnett square with 4 columns, 4 rows. And if you were to solve it, which is done here for you, you would get that 9-3 to 3-1 distribution, right? The other approach to what we just saw is to say, well, if they're fully linked, I know that in my dihybrid, somehow the dominant alleles that came from one parent, from this first parent here, are going to always travel together into gametes. So they're going to go into gametes together. And the recessive alleles will go into gametes together. And they will never interchange. So there will be no recombinant gametes. So if these dihybrid individuals are producing only parental gametes, you can reduce the Punnett square to a 2 by 2. and you end up with 2 columns, 2 rows, and when you solve it, you get the 3-to-1 phenotypic distribution. If the parents, instead of being 2 dominants and 2 recessives, had been 1 of each, when you do that, you end up with a Punnett square that will give you a 1-to-2-to-1. You can try it at home with this example. Great. I asked you about the phenotypic distributions, and you answered that. But now we need to apply it to the problem that we had at hand. And that was that progeny of 380 individuals. So how do we transform these things, 3-to-1 or 9-3-3-1, to a total of 380? Well, we just say, okay, that's my total, 380. What I'll do is I'll multiply 380 by 9/16, 3/16, 3/16, 1/16, and that's going to give me the actual relative amounts of plants of each phenotype. And I can do the same here, 380 times 3/4 and 1/4. And if we do that, these are the numbers that we get. So if these 2 characters are fully linked, like those rice or wheat plants with the long yellow grains or round and brown, you would expect roughly 285 plants producing purple flowers and long grains, and only 95 of them red flowers and round pollen. And then if they were fully unlinked and behaved completely Mendelian way, then you would get that approximate distribution of plants in the F2 progeny. But when these people did this, they found that the number fell somewhere in between, right? With a little more parental individuals that they would have expected by fully Mendelian, and some recombinant, but not as many as you would have expected by a 9-3-3-1. Okay? So the people who described this are named Bateson, Saunders, and yes, the Punnett of the Punnett squares, the person who invented the Punnett squares, found this, and they call this partial linkage. So to go back to our list of possibilities, when there is no linkage at all, half the gametes are like the parental, half are recombinant. When there is full linkage, all the gametes are parental, no recombinant. When there is partial linkage, most gametes are parental, and then some gametes will be recombinant. And I'm vague here because partial linkage can go anywhere between no linkage at all and full linkage, right? Partial linkage is defined as somewhere in between. All right, if not, let's take a look at a different example. And the reason I wanted to introduce this example, it's present in many of your sources. And we will use this example of the mice on Thursday when we talk about mapping a lot. So we're going to go through that different example. Notice that what we're doing here is crossing 2 pure breeding parental lines. There is this first line with mice that have a brown coat and a short tail, to mice that are white and have a long tail. And it turns out that the dihybrid are brown mice with long tails. So what's different about this particular crossing scheme compared to the previous one is that the pure breeding lines are not double recessive and double dominant. We have a pure breeding line that shows the dominant trait for coat color, brown, but the recessive trait for tail length, short. The other pure breeding line, parental, is dominant for tail length, long, and recessive for coat color, white. Okay? So they're mixed and matched of sorts. So far, so good? Yeah? Cool. So how would you determine if these 2 loci, A and B, are fully linked, not linked at all, or partially linked?
The next generation.
The next generation. So meaning, how would you obtain your F2?
So like if it was a 9-3-3-1, you could say it might be a
Excellent,
It's like
excellent. So
majority.
basically, what you're saying, you're proposing is to run a dihybrid cross of these F1 individuals. Cool, like we just did with the flowers. You do several of these dihybrid crosses. With mice, it's not like with plants that you can self-fertilize them, but you can take a brother and a sister, and another brother and another sister, and so forth, have them mate one another, and you can produce a lot of progeny, and then you ask the question, are they following a 9-3-3-1, or are they closer to a 1-2-2-1? Okay? Because here we have one dominant and one recessive. Again, you can try it at home. So that's one way. What's another way of determining if loci in a particular hybrid individual are fully linked, not linked at all, or partially linked? How can we, quote unquote, query gametes for their allelic composition? That keyword that starts with a T. A test cross, right, the test cross. Oh, and before we get into the test cross, this in a way I mentioned, but I'll give it a name now. New concept alert. So whenever you have the two dominants in one parent and the two recessives on the other, when you have the dominants going together and the recessives going together, we call that a coupling configuration. Where that name comes from, honestly, don't know. And whenever you have, like with these mice, a recessive for one traveling with the dominant for the other, and vice versa, the dominant for the first traveling with the recessive for the second, we call that repulsive configuration. And again, this concept will crop up mostly in the way that questions are written, right? So a question may say, you have a cross between a pure breeding parent in coupling configuration or in recessive configuration, and immediately that tells you if they're both dominants or both recessives struggling together or one of each. Anyway, so back to the idea. If we run a test cross, what we would be doing is crossing those mystery hybrid mice to try and determine if these laws are linked or not to double homozygote recessives. That's the definition of a test cross. You cross it to an individual that is very special, that has nothing but recessives for more or one loci. In this case, we're dealing with locus A and locus B, so we cross it to a double homozygote recessive for A and B. If this dihybrid individual, if the loci A and B are fully linked, it means that this dihybrid individual can produce only two types of gametes, the parental gametes. And these parental gametes came in these two shapes, recessive for A, dominant for B, or dominant for A, recessive for B. I'm going to give you a second for that to sink in, but essentially, we in the previous examples with the plants, we always saw the dominants going together and the recessives going together. Here we have an example of one of each, because in those mice, originally, they were in repulsive configuration. So if these loci were fully linked, you would imagine that from the parental generation, alleles for these loci always travel together. And if there was a mouse that was recessive for A and homozygote dominant for B, then they would always put in gametes a recessive for A and a dominant for B. And this dihybrid F1 generation will do the same. Will make gametes with a recessive A, a dominant B, or the parental, the allelic combination, the other parental gamete, that would be dominant A, recessive B. Okay? If you test cross those to an animal that can produce nothing but double recessive gametes, what you would see is that 50% of the progeny is like one parent, short tail and brown coat, and the other 50% is like the other parent, long tail but white coat. Why? Because the only two types of progeny that could have been produced came from this. Let's run this super mini Punnett square here. This is from the F1 dihybrid. This is from the double homozygote recessive. And all you get is, so this is the mouse with short tail but brown, and this is the mouse with long tail but white. That makes sense? So far, so good? So this is just a prediction. If they're fully linked, I expect to see in a progeny of 120, I expect to see about half and half for each phenotype. If they're not linked at all, these are not going to follow the 9331 because this is not a dihybrid cross. This is a test cross. And if you recall, in a test cross, we would get a 1-to-1-to-1-to-1 distribution of phenotypes because we're testing the different gametes that these dihybrid can produce, which now can come in all allelic combinations, to one type of gamete produced by the double recessive. Right? I didn't write it there, but I'll write it again here. These gametes. So you're not getting a 9331, you're getting a 1-to-1-to-1-to-1 phenotypic ratio because you're testing it against double recessives. And once again, if the progeny is 120, you would expect that you get all possible phenotypic combinations in equal ratios, 30, 30, 30, 30. If they're partially linked, you know that it's somewhere in between. We can't really put numbers into it in advance. All we can say is if they're partially linked, then the parental phenotypes are going to be somewhere between 30 and 60. 30 is the least that you can get if they're completely unlinked. 60 is the most that you can get if they're fully linked, so they're going to be somewhere in the middle. The other parental will be somewhere between 30 and 60, and then the recombinants are going to be somewhere between 0 and 30. And lo and behold, when we actually do the experiment, we get, let's say, those numbers. Super quick, because these may crop up. I don't think there are test questions related to this, but it's a concept that shows up in some of the textbook material. How do you determine if that follows a 9331 or if it's more some sort of partial linkage? Like, how do you decide if these can approximate a 9331? Divided by 7, or 16. Yeah? So you divide this number by 16 and then multiply by 9, 3, 3, 1, and see if the numbers fit. Okay, let's say that we do that, and for argument's sake, we get 52, 38, 38, and 6, whatever, right? How do you determine if these numbers are different enough that you're really deviating from the 9331, or it's just by chance? In general, how do we determine if some observations that we make are likely due to something real, something meaningful, or it's just by chance? What do we do? What do we do? What do we use? Start with an S. Ends with statistics. This is across all disciplines, biology, all life sciences, physical sciences. Statistical significance. You're trying to determine the statistical significance. What we're really saying there is we use mathematical formula that tell us what is the probability that what we see is just by chance, right? If I toss a coin and I get heads, someone could come and say, oh, that coin has 2 heads because you tossed it and you got a head. You would say, well, no, it's just one toss, right? Let me toss it several times. You'll see that I get half the time heads, half the time tails. Likewise, if we do this and we get whatever distribution of different phenotypes, someone might rush to say, oh, see, you're deviating from the 9331. You're not getting exactly the numbers that you were expecting by a 9331. But then someone might say, well, no, I mean, it's not, none of this is written in stone. It's just probabilities, right? So maybe you deviated a little bit one way or the other, just like if you toss a coin 4 times, chances are you're not going to get exactly 2 heads and 2 tails, right? Yes? So same here. So what we would do is apply a statistical test, and that statistical test has a name. It's called the chi-square test. And you may have heard about it before. Okay, the chi-square test, or a more modern version of it is the Fisher's test. Okay? So let's say that we run the test and it says, yeah, I think that the chances that you get these numbers by chance are extremely low. So there's a good reason to believe that it's not like they just deviated a little from the 9331 due to chance. It's more likely that they deviated for real from the 9331 because there is partial linkage. Okay? Yeah? Cool. So let's discuss briefly the cell biology behind no linkage whatsoever. We've seen this before. We will go back to the example of the peas that Mendel worked with for some familiarity. But the idea here is often when loci, not always, but when loci are not linked, it often happens because they live in different chromosomes, right? And when they live in different chromosomes, when the dihybrid individual that has one chromosome with the dominant for one and a chromosome with the recessive for the same locus, but then a different chromosome with the dominant for the other locus and a recessive for the same locus, the hybrids will replicate the DNA, then the chromosomes will align across both sides of the metaphasic plate, and that's where this random orientation, the random relative orientation of the chromosomes, will lead to the random distribution or independent distribution of alleles in the gametes. We discussed this when we talked about Mendel's second law, that you don't see it in only one meiosis. You have to look at this across several meioses, and then you will see that in some cases, the two dominants will end up on the same side of the metaphasic plate. In about the other half of the cases, they will end up on opposite sides. And that's how the two dominant alleles are unlinked from one another. They're not following each other. Each of them is doing its own thing. The reason for that is here, that the orientation of the homologous pairs is random across both sides of the metaphasic plate. So after meiosis 2, half of the gametes are going to have these allelic combination of both recessives, and we call, or allelic combination of both dominants, we call those the parental. And then the other half will have the recombinant. If this is a little too much, it's just a refresher. We talked about this. We have some slides dedicated to this in previous lectures. We're just revisiting the cell biological basis of most of non-linkage that exists, because loci live in different chromosomes and therefore are going to be distributed into gametes independently from each other. When loci are located in different chromosomes, they will assort their alleles into gametes independently, giving rise to all possible allelic combinations. This is because homologous chromosomes orient each other on each side of the metaphasic plate independently from what other homologous pairs do. What's the cell biology behind full linkage? Well, the easiest way to understand full linkage is to imagine these loci existing on the same chromosome and really close to each other, right? So wherever one goes, the other one goes. So in this case, we go back to the example with the rice grains. So in this case, we have the two recessives traveling everywhere together and the two dominants traveling everywhere together. Notice how now, when it comes to those two loci, the locus S and the locus B, even though the chromosome carrying those loci may flip orientation in one meiosis after the other, they will always be together. So these chromosomes flipped orientation. So on the left, we have blue on top, pink on the bottom. On the right, we have pink on top and blue on the bottom. But that didn't change the companion that each of these alleles had, because they're going everywhere together. So even though the chromosome is flipping on one side or the other of the metaphasic plate, that flipping could not separate these two alleles. And the two dominants are going together, the two recessives are going together. What would we say about these two loci in this particular scenario? Are they in coupling configuration or repulsive configuration? Coupling, yeah, like coupling. Coupling, yeah. they're like, I don't know, to me, the best way to remember is they're like a couple, right? So after meiosis, and here's something that I want to emphasize. So after meiosis, you'll notice that half the gametes, when it comes to the loci S and B, half the gametes have the two recessives, right? So I'm going to mark them here. So this half has the two recessives. This half has the two dominants. When it comes to these two loci, it's not like all gametes are the same. Half of them are going to have both dominants, half of them are going to be both recessives. So they're not all the same, but they're all parental, because you could see both of those gametes in the parental generation here, right? So from an S and B perspective, there is 100% parental gametes, 0% recombinant. But the reason I threw locus A there was to emphasize the fact that, once again, when it comes to other loci, these two that are fully linked to each other, they are completely unlinked from other loci. And to illustrate that point, I threw locus A there on a different chromosome, and now the flipping of the homologous pairs matters, right? And even though the A locus was hybrid in that F1 trihybrid generation as well, now you see that when it comes to the SB pair relative to A, you get 4 combinations. You get the two alleles with the two recessives with a dominant A, but you also get the two recessives with a recessive A. You get the two dominants with a recessive A, but you also get the two recessives with a dominant A, the two dominants with a dominant A, right? Now, when you go back to what the parental gametes look like, you notice that there is 2 new ones. All 3 recessives and all 3 dominants did not exist among the parents. Neither of the parents could produce gametes that had all 3 dominants or gametes that had all 3 recessives. So those 2 new gametes are new. They're recombinant. And they actually account for half of the gametes produced by that individual. So when it comes to S and A, for instance, we go back to fully unlinked, 50% are parental gametes and 50% are recombinant. So the fact that locus S is fully linked to B and produces nothing but parental gametes says nothing about how it behaves with respect to locus A. It's a different locus, different story. Okay? And last, what is the cell biology behind partial linkage? And I know we've been bringing in crossing over throughout the weeks to always kind of rule it out and push it aside, right? We said, does crossing over affect Mendel's first law? No, it doesn't. Does crossing over affect Mendel's second law? No, it doesn't. We went through several examples of how you can have all the crossing over you want. It won't affect the equal segregation of alleles for one locus into gametes or the independent segregation of fully Mendelian loci that are unlinked into gametes. But finally, crossing over comes to the rescue, and it will help us explain partial linkage. So the way this works is, when you have partially linked loci, like let's say we go back to locus A and B in these mice that didn't show 60-60 or 30-30-30-30, they show something in between that we determined statistically was significant. So now that we've established the partial link, what's going on? Well, we know that these two loci will live in the same chromosome, and that's why they're partially linked. But they can also become separated once in a while. That's why they're only partially linked, if that makes sense, right? So they're linked because they live on the same chromosome, but they're only partially linked because there is something that can separate them. So when you have partially linked loci, and these individuals make gametes through meiosis, most gametes will be parental because in most gametes, these two loci will be traveling together. They are linked to some extent. In some meiosis, what's going to happen is when the two homologous chromosomes face one another during prophase 1 of meiosis 1, they will undergo crossing over. Two chromatids from each of the homologous chromosomes will form this thing that we call a synapsis, and they will exchange genetic information such that they swap a portion of the chromatid with the other homologous chromosome. So you end up with a chromosome that has a chromatid that is like the original, a chromatid that is mostly like the original with a little bit of that it took from the homologous pair, and then the other chromosome, same thing. One chromatid that is like the original and the other one that is mostly like the original with one little bit that was swapped, exchanged. So those meioses in which there was no crossing over will give you parental gametes, because the loci in those cases, or the alleles for both loci, were traveling together. And they've been traveling together since the parents, so they will end up in the gametes like they came from the parents, so they're parental gametes. Here is where crossing over throws a bit of a wrench in the process, and now these alleles that were traveling together from the parents swap, and they form some recombinant gametes. Now, there's a number of things that we need to emphasize about crossing over, which will help us explain partial linkage. The first one is, even when there is crossing over, notice how when there was a crossover event, there is still chromatids that retain the parental configuration. There is still two chromatids that are not involved in the crossover event. They're untouched. I emphasize that these two chromatids are like the original. And in those original chromatids, the alleles are going to continue traveling the way that they've been traveling from the parents. And they will give rise to parental gametes. Whereas only those that actually suffer the crossover event are the ones that will generate recombinant gametes. The absolute extreme scenario would be that every meiosis has a crossover event between these two. Unlikely, but let's say that every meiosis has a crossover event between those two. Even in that extreme scenario, half the gametes would be recombinant, half of them would be parental. But that's an extreme scenario because crossover events are rare. They happen, but they're rare. So if anything, most meiosis would be more like this. And the more rare meiosis in which there were crossing over events, half of the gametes will be parental anyway. That's why you end up with mostly parental gametes, some recombinant. Okay? Cool, but how many? How do we calculate how many recombinant gametes there were? So there is this concept called the recombination frequency, and it's just a calculation. And you will need to learn this formula because you'll have to apply it to problems, and sometimes there may be questions that simply ask you about this formula. The recombination frequency is calculated as the number of recombinant gametes versus the total number of gametes. Now, when you're dealing with a test cross, you know that looking at the phenotypes in the progeny is almost like looking at the allelic composition of the gametes, because the gametes that the double or triple homozygous recessive provides have nothing but recessive alleles for those genes. So by looking at the phenotypes in the progeny, you're essentially looking at the allelic combination of the gametes produced by that F1 dihybrid or trihybrid. The reason I say that is because we can say here number of recombinant gametes over total number of gametes, or we could say number of recombinant phenotypes versus total progeny. So I'm going to write it there. Number of recombinant phenotypes versus total progeny. Either way, here you would do 13 plus 17. That's either the progeny with recombinant phenotypes that you didn't see in the parents, or the number of gametes that you know gave rise to these recombinant phenotypes over the total. and that gives you 0.25. So what we say is that the recombination frequency between locus A and B in these mice is 0.25 or 25%. We'll take it up from here on Thursday.